In this video we will discuss about matrix stiffness and tumor proliferations. So basically we will discuss this stiffness and proliferations from 4 different aspects. First one is about the stem cells. So in the stem cells, the stiffness not only increases the stemness of these cancer stem cells, but it will also helps in the interactions of this Id1 and Id3 through this TGF beta, and this activations of the Id1 and Id3 leads to the more tumor initiating probabilities through this GIC. Along with that, it will also helps in this myeloid, these MSCs formations, and this will also helps in the increase in the matrix stiffness. And in terms of this tumor invasion and proliferations, this matrix stiffness not only helps in the this tumor proliferations through this Ki67, can be monitored through Ki67, but the tumor compositions also plays an important role to determine how the tumor proliferate and invade. And some of the examples we have seen in the previous video that it will lead to the phosphorylations of this FAK SRC pathways, and that will lead to this ROH or ROCK inhibitors pathway activations, and that basically plays a main mechanosensors between tumor and matrix, and that in turn leads to the tumor invasion and proliferation characteristics as the stiffness around the matrix increases. And next part is about the tumor metastasis. So here one of the new things that I find out is this BMDC plays an important role in this tumor colonizations at distant organ. So in that, what how they helps in this tumor colonizations at distant organ is through these angiogenic factors or other growth factors. So angiogenic factors, what happens, it will create a new blood vessels through which these cancer cells can migrate into the endothelial cells and can metastasize. Along with that, the stiffness will also leads to the localizations of this 2:1, and that in turn helps in the metastasis of the cancer cells. Along with the 2:1, this EGF, E2F3 will also be localized. This will also lead to the metastasis of this tumor cells. And the last main factor or main relation between stiffness and tumor is about the EMT. So in the EMT, what happens is there's mesochemical characteristics such as N-cadherin goes up, and also the snails goes up, and the E-cadherin markers such as epithelial markers such as E-cadherin goes down, and this will leads to the these things will leads to the EMT. This EMT will leads to the release of cancer associated fibroblast, and that will leads to the increase in the ECM stiffness, and as the ECM stiffness increases, this will finally lead to the YAP and TAZ nuclear localizations, and this YAP TAZ nuclear localizations will cause upregulations or will finally results in this more EMT changes, that's epithelial to mesenchymal transitions. And in this case, the FAK SRC activations will also plays a role, and along with that, the upregulation of snails, which is a mesenchymal marker, will leads to the decrease in E-cadherin, which is a epithelial marker. So this is how the 4 different main categories where stiffness and tumor proliferations can interact with each other. And in the case of like EMT, this could create a cascade effect that will lead to the further strengthening or further stiffening of the ECM or extracellular matrix. One thing that is not mentioned here, I guess, is about the TGF beta effect. So with the increase in the EMT, it will lead to the autocrine TGF beta effect. That is, TGF beta will lead to the more EMT transitions, and that will cause the more TJ beta formations, and this TJ beta will also affect the EMT transition in the nearby cells. So this is basically called the TJ beta autocrine EMT, which is epithelial to mesenchymal transitions. So I hope you understood how this stiffness and tumor proliferations plays a role in these 4 different categories broadly. If you need to understand more about this relations in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.